Okay, so yes, I will talk about retinitis pigmentosa and more largely about retina and the programs on cell therapy that we have developed in the lab. And just to give you a brief insight on what we're talking about. So the retina is the photosensitive layer at the back of the eye. Is a very, very organized layer, as you could see here, with many cells that has to be put in the right order and right direction. And among those cells, the cells that are very important for the sight is the photoreceptors, the cone and the rods that are just there. And at the very back of the eye here, you have a layer of retinal pigment epithelial cells. And those cells are really the babysitters of the photoreceptors. So they provide nutrients, they clean the photoreceptors, they are very supportive cells. And actually in some disease like retinitis pigmentosa, in about 5% of retinitis pigmentosa, when those cells are dysfunctioning, not providing the right function, then the photoreceptors will die. And it's also the case in the much more frequent disease, which is the age-related macular degeneration, the AMD, wet and dry form, where the same cells are dysfunctioning, eventually dying, and then the photoreceptors will die, and then the patient loses sight. So the idea we developed in the lab was actually to differentiate some RP cells from embryonic stem cells, from pluripotent stem cells. We do that with human pluripotent stem cells, embryonic and iPS. And then to generate a little patch, because you saw that in the retina, RP cells are very organized. They are forming a layer. So we generate a patch to reconstruct this layer. And the idea in the first program that we actually developed was really to delay or even to stop the degeneration of photoreceptor. So we developed this product, and because I don't have much time, I will have to cut a very long story very short. So we developed this little patch that you could see here, which is a SIM card format to keep the orientation of the cells. The patch is about 5 to 3 millimeters, and it's embedded in gelatin to actually keep also to protect the cells, to give some elasticity to the patch, to be able to inject the patch under the retina. And we also developed some tools and some specific medical device to prepare this patch without using scissors and forceps, and to provide like a clé en main this patch to the surgeon. So we did that in close collaboration with the Institut de la Vision in Paris, with the Hôpital 1520, Hôpital Saint-Louis, and the AFM Téléthon. We perform all the steps to prepare, so it's more than 10 years of work. So we did all the steps, so by concepting and having the right protocol to obtain the right cells, so really work on quality controls, performing new quality controls and improve the quality controls, but also we did all the preclinical animal validations needed. I talked about this medical device, and we started actually the clinical trial in 2019. It's a phase 1/2 clinical trial. So the first point is really to tackle the safety of this product, so to look at tolerability, safety of this product. And actually we were able, despite of the pandemic and the COVID-19, we were able to graft 7 patients that are followed in the Hôpital 1520. And the PI, the surgeon, was the Dr. Stéphane Bertin. And those patients are actually specific mutations, but they have 2 different mutations. So it's a cohort of different patients that we followed in this first clinical trial. So where are we now? So we're following this patient, but in the same time, we are trying to improve this patch by trying to automate the scale-up of RP production in order to go to monogenic disease, rare disease, to eventually a more frequent disease, the AMD. The AMD is about 2 million patients just in France. So, it's a lot of patients. So we worked a lot on automation, and we have validated actually the protocol that allow us to produce the RP in different automate. And from 1 million, we obtained 16 billions of cells with exactly the same quality than the one that was done manually. And the second aspect is also to work on cryopreservation, because we know how to cryopreserve the cells, we know how to cryopreserve the membrane, but now we're trying to cryopreserve actually the entire patch to have a sort of a off-the-shelf product that would be easily to go to the patient and to also reduce the cost of the production of this patch. And finally, so what are the perspectives beyond this RP patch? Of course, is to add the other types of the cells that are in the retina, so adding the photoreceptors on the patch. So it's a work that we are performing with, in collaboration with Institut de la Vision and also with some physicists in Paris-Saclay, because we need matrix, we need scaffold to put the cells together and to do some tissue engineering. To have more complex tissues to actually cover the entire spectrum of retinal dystrophies. We're also working a lot on trying to escape immune system, so working on getting more some immunotolerant cells by different approaches. Working also combining eventually extracellular vesicle therapies to maybe decrease the inflammation, prepare the host environment, and also trying to improve the quality control by using some artificial intelligence. And just to finish, this is the pipeline that we are addressing. So we have a strong pipeline addressing different types and stages of all the retinal diseases. So in monogenic disease, so in retinitis pigmentosa, the patch we have developed cover about 5% of the disease. So we're working on trying to develop products for the other 95%, but also developing a more complex patch for more frequent disease like the AMD at different stages. And just the last slide, but maybe the most important, is to acknowledge all the people that have worked since more than 15 years now on this program, and also the patients that are participating to the clinical trials. Thank you.
Thank you very much, Christelle.